Hi, everyone. I just wanted to start by thanking Jacinta and HealthCert for inviting me to present at this masterclass. I do feel very honored to be taking part, and I apologize that I can't be there in person. My presentation is on the gut-skin connection in skin cancer. And the gut-skin connection is a topic that I've been exploring for almost 15 years in relation to autoimmune and metabolic diseases. And over the last 8 years, also in relation to cancer out of personal necessity, which I'll explain in a couple of slides. So I'm a cosmetic and nutritional medicine practitioner, and I've had an interest in nutrition since my early teens when I started to experience a number of unexplained internal and external symptoms. And it took almost 20 years before I got a diagnosis, or actually multiple diagnoses. But during that time, as I went through my undergrad and medical degrees, I researched and trialed many different nutritional interventions and was eventually able to get my symptoms under control with nutrition alone. So by the time I completed my GP fellowship, I knew that I wanted to help patients achieve the same results, which is why I went straight into nutritional and cosmetic medicine. And since then, I've founded a company called Bio-Alai, and I've developed a health system that combines personalized nutrition plans with a complementary medicine that practitioners can offer as an adjunct to their services. So this is my disclosure, but I won't be talking about my products today. What I will be talking about is the science, or at least a very small part of the science on which they were based. So, what I'll start with today are the hallmarks of aging, because these overlap with the hallmarks of cancer and they form the treatment targets of nutritional interventions. I'll then expand on the gut microbiome and its connection to skin diseases, but I'll focus on immune function and epigenetics in relation to melanoma. And then I'll look at how nutritional interventions can support the key pathways involved and improve efficacy and outcomes for those on immunotherapy. But before I move on to the science, I just wanted to talk about the reason why I was actually so keen to present at this masterclass. In 2017, my husband was diagnosed with locally advanced tongue cancer. We were told the prognosis was good for the type of cancer that he had. So he underwent a course of chemo and radiotherapy, and of course, alongside a number of complementary treatments that were approved by his oncologist. And the treatment appeared to be successful. But 1 year later, the cancer had metastasized to his bones. And this is what you see in the scan here. We were told at that point that he qualifies for immunotherapy and that would give him an extra 12 to 18 months. And so he was started on it straight away. And that's when I started to really look at the research into nutritional interventions to support him through it. And I came across this article, which had been published 2 years earlier. It was from a study that was conducted at the MD Anderson Cancer Center, and they found that melanoma patients responded better to immunotherapy if they had a diverse microbiome with particular species present. So I then went and looked at all the research around this and developed a complementary plan for my husband. And I'll go back to his case at the end of the presentation, but I'll now present the science that explains why a diverse microbiome is the single most important aspect of human health. And it can actually be regulated quite simply by what we eat. So my hope is that my presentation motivates you to discuss nutrition with your patients, as it really may improve their responses to immunotherapy and even to any other treatment they may undergo for any condition at any point in their lives. So, by far the most significant finding in the last 20 or so years is the role of the microbiome in regulating human health. A diverse microbiome supports health, whereas a dysbiotic microbiome underlies chronic disease and aging. And the hallmarks of aging are the degenerative molecular processes through which dysbiosis appears to drive the decline. And from a more mechanistic view, the gut microbiome is a key regulator of immune function. And so dysbiosis produces a chronic state of inflammation in the body, which changes the epigenome, disrupts mitochondrial function, impairs autophagy, nutrient sensing and also intercellular communication. And the oxidative stress that accumulates goes on to damage DNA and telomeres, and this leads to the accumulation of senescent cells and depletion of stem cells. So I'll expand on some of these as we go through the presentation. So the effects of the hallmarks of aging on aging cells versus cancer cells differs. In aging cells, all of the hallmarks lead to decline, whereas in cancer cells, some of the hallmarks will promote tumor growth while others have an antagonistic or neutral effect. So what I'll focus on are the tumor promoting hallmarks, which are dysbiosis, inflammation, epigenetic alterations, mitochondrial dysfunction, and impaired nutrient sensing, which contribute to genomic instability and tumor progression. So both in the skin and systemically, the greatest issue is the consequences of these hallmarks on the immune system, because immune cells are our defense against cancer. But keep in mind that all cells are affected by these same processes. So dysbiosis and inflammation produce changes to the epigenome, which what regulates the expression of our genes, and that includes the expression of DNA repair and endogenous antioxidant systems. And inflammation also impairs mitochondrial function, so energy production declines and free radicals increase. And the resultant oxidative stress basically impairs cell function and produces senescent cells. These are also called zombie cells because they remain alive, but they don't function. Now, senescent cells secrete specific inflammatory cytokines that convert surrounding normal cells to senescent cells, and this drives the aging process. And this is what's commonly referred to as inflammaging, but it doesn't just occur in advanced age, because senescent cells are found in many common conditions and skin conditions like psoriasis, acne, eczema, and skin cancers that we know can occur at any age. In relation to melanoma, T cell senescence is linked to poor response to immunotherapy, because immunotherapy relies on functioning T cells to attack cancer cells. When immune cells become senescent, we not only lose our immune surveillance and protection, but inflammation worsens, and this produces a vicious cycle. And chronic inflammation is the cause, and it's the key driver of tumor initiation and progression. So inflammation from any cause, whether it's a chronic viral infection, autoimmune disease, or lifestyle and environmental exposures, not only increases our risk of cancer, but also worsens outcomes. And these underlying processes are often overlooked in those undergoing immunotherapy. So research is looking at how we can intervene, and there are a couple of treatment targets, which of course include inflammation, but also its downstream effects, which are impaired nutrient sensing, mitochondrial dysfunction, and epigenetic changes. And these ultimately disrupt cellular homeostasis and lead to T cell senescence. So the goal of nutritional interventions is to reduce inflammation in the body and target each of these processes to restore cellular homeostasis. And these are actually the factors that are now recognized as the main causes of chronic inflammation and almost every chronic disease. And as you can see, it's not rocket science. Our diet and lifestyle and the increasing environmental toxins that we're exposed to are causing inflammation that's driving chronic disease. And the effects of these factors are largely mediated by their impact on the gut microbiome and the epigenetic and metabolic consequences that arise as a result. So this discovery really provides a safe framework for addressing the true causes of chronic disease. And as you can see, there are diet and lifestyle factors here, but I'm going to focus on nutritional interventions, because what I've found in my practice is that by starting with dietary modifications, everything else seems to fall into place. And that's because sleep improves, energy levels improve, and that makes it easier for patients to start regular activity, and brain function and decision making improves. So patients find it easier to make choices or decisions that are consistent with their health goals. So nutrition is important simply because with every meal, we change the gut microbiome, and we now know that the gut microbiome is central to human health. Our body has more microbial cells and genes than we do human, and their microbial metabolites regulate and communicate with every organ and system in the body. Everyone has a unique microbiome, but the key features of a healthy one include a diverse number of species with higher commensal species, less pathogenic species, and the main bacterial phyla are kept within certain proportions relative to each other. That's what you see here in the pie charts. And this will vary between microbial communities in different parts of the body. So our healthy species need dietary fiber to survive. They have over 100,000 genes for the digestion of dietary fibers, whereas humans only have 17 genes for this role. And their digestion of fiber produces over 200 metabolites that support all aspects of our health. The main metabolites they produce are called short chain fatty acids, and butyrate plays the most significant role in regulating immune function. But the microbiome also produces various enzymes and vitamin K and the whole spectrum of neurotransmitters and B group vitamins as well. So in the gut, short chain fatty acids maintain the integrity of the gut wall, which is a barrier to the outside world, just like the skin is. And they also regulate innate and adaptive immune cells to produce an anti-inflammatory response, which, along with microbial metabolites, is transmitted through the bloodstream to distal organs where they regulate metabolism, hormones, and all aspects of brain function and skin health. So this is how they communicate with keratinocytes and melanocytes and the skin's microbiome and immune cells. The Western diet, on the other hand, is high in saturated fats and sugars and a lot of additives and very low in fiber. And it produces a state of dysbiosis where there's an increase in pathogenic species like E. coli that produce harmful metabolites. And one of these is called endotoxin. An endotoxin breaks down tight junctions between enterocytes and it increases the permeability of the gut wall, allowing endotoxin and other gut antigens to cross through the gut wall and activate immune cells to produce a pro-inflammatory response. And 80% of all of the immune cells in the body are located along the length of the gastrointestinal tract. So this is where the immune system is primed. And the inflammatory signals, along with microbial or more harmful microbial metabolites, go on to disrupt metabolism and blood sugar control. They change our appetite, our mood. They impair our decision making and disrupt skin homeostasis as well. So this is how inflammatory signals that originate in the gastrointestinal tract disrupt skin homeostasis. They basically produce an inflammatory metabolic and neuroendocrine response within the skin, and they also disrupt the blood-brain barrier and produce inflammation within the brain and activation of the HPA axis or stress response. And this in turn activates the skin's own stress response. So this is called the gut-brain-skin connection. And this is why skin conditions tend to flare during times of stress. So there's a lot of clinical evidence for this gut-skin connection that has been quite well documented. About 25% of people with celiac disease or inflammatory bowel disease also have concurrent dermatological conditions, and about 46% of those with rosacea have a condition called small intestinal bacterial overgrowth, or SIBO. An antibiotic treatment of the SIBO results in resolution of the rosacea, so that's a very clear gut-skin link. And similar links have been found for almost every chronic condition, including obesity, cardiovascular disease, and cancer. And the pathologies are mediated through inflammation and its impact on metabolism and neuroendocrine function within the end organ or tissue or system. Now, there are a couple of things I just wanted to highlight here. The first one is the microbial metabolite butyrate, which is a substrate for the mitochondrial chain in enterocytes. So it supports the gut barrier, and it's really important in maintaining the integrity of the gut wall. And it's also an epigenetic regulator that potently downregulates the inflammatory response in immune cells. So high butyrate levels have been linked to a range of health benefits. The next one is endotoxin, which activates a group of pattern recognition receptors called toll-like receptors that initiate a pro-inflammatory response in immune cells, and this has been linked to almost every chronic condition as well. And the final one is a bacterial species called Akkermansia muciniphila, which is crucially important in regulating and regenerating the mucus layer of the gastrointestinal tract. So it kind of provides a first-line defense against antigens, but it also improves immunosurveillance and defense against cancer. So it improves immune function. And it was one of the species that was common in responders to immunotherapy in the MD Anderson Cancer Center studies that I initially read about. Unfortunately, this particular bacteria doesn't come in a probiotic form, but certain prebiotics can increase its numbers, and I'll talk more about that later. So researchers found that lower diversity and changes to certain bacterial species appear to be more prevalent in those with melanoma. In this particular study, they also looked at microbial changes in basal cell carcinoma and also ease of tanning. So there are specific microbial patterns that appear to be linked to these conditions. And if we take a closer look at how microbes regulate immune function, our commensal species release, in addition to the microbial metabolites they also release certain molecular patterns that downregulate inflammation and produce a state of immune tolerance, whereas pathogenic species produce molecular patterns that are, again, recognized by toll-like receptors, and this leads to activation of the inflammasome and then activation of a specific inflammatory response. And different microbial species will activate a different T cell response to produce either an allergic, autoimmune, or basic inflammatory response, depending on genetic predisposition. And each of these responses has been linked to specific skin conditions, as you can see here. And it's also been linked to almost every chronic condition. And all of these responses can coexist, and all of them have been linked to tumor initiation and progression. So that's why it's so important to normalize immune function. And it's actually not that hard to do, because processed food is the cause of dysbiosis in everyone. But because everyone has a different baseline microbiome and a different epigenome, and also a different genetic predisposition, their immune response to processed food will differ. So those without a genetic predisposition will simply have a chronic inflammatory response that can be quite mild, whereas those with a genetic predisposition to allergies or autoimmune disease are likely to experience ongoing symptoms as a result of the inflammatory response produced by these or initiated by these food compounds. So everyone's affected by processed food, but those with a genetic predisposition to allergies or autoimmune conditions can also be affected by foods that are generally considered healthy, like whole grains and legumes, and even certain fruits and vegetables. So these individuals do much better with a personalized diet. And if it's implemented correctly, their tolerance to these foods tends to improve as microbial diversity increases, because a diverse microbiome is more resilient to both the microbial and the epigenetic changes that these food compounds produce. And so that brings me to the epigenome, which is a collection of protein and DNA modifications that regulate the expression of our genes. So our genes are fixed, but the epigenome can be modified by what we eat and how we live our lives. And two really important epigenetic modifications are methylation and acetylation, which upregulate molecules that repair breaks in DNA, scavenge free radicals, and activate autophagy to remove senescent cells. With age and various cumulative exposures, methylation and the expression of repair genes reduce, and in mice, these epigenetic changes are linked to skin aging and hair loss. The one way we can support the epigenome is by increasing dietary fiber, because this increases butyrate. And butyrate is it's called a histone deacetylase inhibitor, which inhibits an enzyme that removes acetyl groups from the epigenome. But it's a dose-dependent factor. So one study found that the benefits occur depending on the dose of or the level of butyrate present. So when it's low, it promoted proliferation in colon cancer cells, whereas high levels led to apoptosis. So 20 to 30 grams of dietary fiber per day is known to significantly increase butyrate levels. So this is a safe recommendation we can make to our patients. So we can also support methylation with certain nutrients that are known as methyl donors. These reprogram the epigenome and change gene expression. And in this study in mice, they found that pregnant mice with the gene mutation that causes yellow fur and obesity produced normal offspring when they were supplemented with these methyl donors. So these nutrients are found in green leafy vegetables and meats, or just in a basic multivitamin form. And certain polyphenols like green tea extract, genistein from soy, and curcumin are potent antioxidants that scavenge free radicals, but these have also been found to increase methylation. And vitamin D is a master epigenetic regulator that influences the expression of 5% of our genes. It plays a key role in downregulating the inflammatory response and improving immune function overall. And it also increases activity of natural killer cells, which have direct anti-tumor activity. Vitamin D also downregulates the hallmarks of aging and improves mitochondrial function and autophagy in all cells. So its benefits extend beyond the immune system. And it's estimated that about 80% of office workers and 80% of those with obesity have vitamin D deficiency. And considering that people are now avoiding the sun to prevent melanoma, it's important to monitor vitamin D levels and supplement if the patient is deficient. So the next process that we need to look at supporting is mitochondrial functions. Mitochondria, as you all know, they're found in almost every cell in the body with the exception of red blood cells. And mitochondria are termed the powerhouse of our cells because they convert macronutrients from food into ATP, which is the cellular currency of energy. And this ATP allows the cells to perform their function. And in the process of ATP production, mitochondria also produce free radicals, which are important molecules because they initiate autophagy, which is the recycling of cellular debris that begins the process of cell renewal. So mitochondria basically maintain cellular homeostasis as long as these two processes remain balanced. With age, and again with these various cumulative exposures, mitochondrial function declines and the balance is shifted. So energy production declines as well, and free radicals build up and they go on to damage mitochondria and various cellular components, eventually leading to mitochondrial dysfunction, which is basically bioenergetic failure. And it's the process that underlies the symptom of fatigue. Now, these are some of the conditions that have been linked to mitochondrial dysfunction. So you can see obesity, autoimmune condition, and fatigue syndromes that are so common in our patients. And this is because energy production declines. Now, all of these conditions increase the risk of cancer, and all of them worsen outcomes for patients with cancer. And over 70% of your patients would have one or more of these conditions. So this study looked at histopathology samples of 12 patients with early melanoma to identify the molecular markers associated with disease recurrence within a 5-year period. And they found that those with recurrence had markers of mitochondrial and metabolic dysfunction, as well as impaired mitophagy, which is the recycling of damaged mitochondria. So that was impaired, and that's an aspect that's really important for cellular homeostasis and to maintain energy production. So mitochondrial dysfunction increases the risk of melanoma recurrence. Now, this study in mice really highlights the importance of mitochondrial function. What you see here are 2 mice of the same age, but the one on the right-hand side had mitochondrial dysfunction induced 2 months earlier. And as you can see, it rapidly aged. And this is the same mouse 1 month after mitochondrial function was restored. So there's a lot of research looking into how we can restore mitochondrial function. And unfortunately, that's led to a lot of gimmicky products on the market claiming to do so. But these are the nutrients here on the left, are nutrients that are essential for different aspects of the mitochondrial chain to proceed. This is just basic biochemistry. And one study that was conducted on ICU patients over an 18-month period where the investigators who were ICU specialists trialed a number of dietary and supplemental interventions, and that included high-dose intravenous nutrients. And they found that by supplying these basic nutrients at recommended daily intakes, as well as early mobilization, was the most effective tool or strategy in restoring mitochondrial function in these patients. And certain polyphenols, which we all know are antioxidants that scavenge free radicals, but these have also been found in animal and human studies to increase mitochondrial numbers and function by activating mitophagy. But it's actually quite hard to obtain the beneficial dose through dietary sources because all but olive oil polyphenols have a very low bioavailability, and many people lack the species that convert them to more bioavailable forms. So this is where supplementation might be beneficial. And just to give you an example, 1 glass of red wine contains 1 milligram of resveratrol, whereas the beneficial dose is believed to be about 1,000 milligrams a day. So you'd certainly destroy your mitochondria if you're trying to get that dose from red wine. And this really is just to give you an idea of how important nutrients are. So these are some of the metabolic pathways in the body that rely on nutrients. So nutrients are essential to life, but according to a study that was conducted by the Australian Bureau of Statistics, I think that it was in 2022, over 70% of Australians consume a diet that's deficient in at least one nutrient, and it only takes one nutrient deficiency to disrupt homeostasis. So if your patients have symptoms or any chronic conditions, it's a good indication that their diet isn't providing the nutrients to support these pathways. And so the right diets, the right supplements at the right doses can make a big difference, at least in the short term, while the patient is optimizing their diet. And so that brings me to what an optimal diet actually is. And there's an overwhelming amount of data on various dietary interventions, and some of it is quite conflicting, so it can be a bit confusing. But by far, calorie restriction, which is the most well-researched of the dietary interventions. And there were 2 long-term studies conducted on rhesus monkeys, which are our closest relatives, and both studies found a range of health benefits with calorie restriction compared to a standard diet. But the findings were actually conflicting with regards to health outcomes and longevity. And the differences related to the age at which the calorie restriction was commenced, the quality and timing of the food supply, and the sex of the monkey. Now, in humans, long-term calorie restriction just isn't sustainable because of homeostatic responses that increase appetite and reduce metabolism. So the focus in research has shifted to intermittent fasting as a more sustainable alternative to calorie restriction. So I'll now look at the science behind intermittent fasting and why these same variables apply. The main pathways that are influenced by intermittent fasting are the nutrient sensing pathways, which determine how the body manages macronutrients. So when we eat, the glucose and amino acids in the meal, along with insulin and growth factors that are produced, activate a molecule called mTOR, which is responsible for cell growth and synthesis. mTOR also inhibits autophagy, which is how our cells recycle cellular debris to rebuild new organelles or cell components, and also to remove senescent cells. So autophagy is important for removing the senescent immune cells that reduce immunosurveillance. When we fast, an enzyme called AMP kinase is activated, and it inhibits mTOR and it activates autophagy and repair molecules. It also increases glucose and fatty acid metabolism, so it improves insulin sensitivity. And this is really important because insulin resistance potentiates inflammation, and also because cancer cells rely on glucose for their metabolic pathways. So clearing glucose from the bloodstream reduces their energy supply. So the general idea is that eating and activation of mTOR is aging, whereas fasting and activation of AMP kinase supports health and longevity. But just like with mitochondria, it's all about balance. And this study highlights that factor. So this image is from a mouse study which looked at the effect of intermittent fasting on wound healing and found that collagen is broken down and stem cells are activated in the fasting state. and then collagen and the epithelium start to rebuild in the feeding state. So alternating fasting with feeding supports skin renewal, and this is actually how it works in almost all cells. The problem is that we tend to eat too much of the wrong food and we eat too often. So mTOR is constantly being activated and autophagy and repair mechanisms are inactivated. So cellular debris and oxidative stress builds up, and this leads to inflammation. And it's inflammation, it's this kind of chronic inflammatory response that leads to metabolic dysfunction and the diagnoses like insulin resistance, diabetes, and fatty liver disease, which are driving weight gain in our patients, but also increasing the risk of cancer and worsening outcomes for them if they are if they were to be diagnosed with cancer. And the more adipose tissue an individual has, the more dysfunctional the immune system becomes, because adipose tissue is an endocrine gland that secretes hormones and cytokines. It's actually one of the largest stores of inflammatory cells in the body. And these inflammatory signals produce metabolic and immune changes within the skin and also systemically, and it causes a vicious cycle. And it also impairs tumor immunosurveillance and increases the risk of chronic infections and autoimmune disease and cancer. And there was one retrospective study of 1,001 patients with melanoma that found that obese patients were almost twice as likely to have metastases in sentinel lymph nodes than normal weight patients. Now, about 67% of Australians are overweight or obese, and they're now being prescribed glucagon-like 1 peptide agonists, and as a result, they're eating less food, so they're losing weight. But the problem is they're still consuming food that causes dysbiosis and inflammation. And unfortunately, the medication itself, which is a potent anti-appetite suppressant, produces changes in brain structure and function that make it impossible to keep the weight off long term when medication is stopped. Now, certainly in those with obesity and a high risk of cardiovascular disease, the benefits of glucagon-like peptide 1 agonists far outweigh the risks, but these patients should also be counseled on dietary modifications to support gut and immune health. Now, intermittent fasting is becoming the diet of choice for most people wanting to lose weight, and these are the more popular forms of intermittent fasting. The first two restrict intake on alternate or specific days of the week, whereas time-restricted eating most commonly limits intake to an 8-hour window each day. So this is what most people are following. And if you look at the overall health benefits of intermittent fasting, these have been found in human studies. They're all based around improving nutrient sensing pathways, which results in improved insulin sensitivity and weight loss, and a number of other metabolic and overall health benefits. But because certain parameters were initially overlooked in these studies, and because everyone differs in their response to dietary interventions, the more recent research is finding that these techniques aren't necessarily translating to health benefits in all. One study found that thyroid hormone levels reduce and cortisol levels increase both with short-term fasting and long-term time-restricted eating. And obviously, this is something that we want to avoid, because low thyroid hormone levels reduce metabolism and decrease mitochondrial function, and persistently high cortisol levels produce inflammation and insulin resistance. And both of these actually ultimately can lead to weight gain. And I do see this in my practice, where patients are following these various diets, are restricting calories, and then they start to experience severe fatigue and gain weight despite eating very little. So these studies are explaining why that might be the case. Other human studies have found a worsening glucose and inflammatory markers with each additional hour of fasting, and one paper reported a 91% increased risk of in cardiovascular related death with long-term time restricted eating. So that's not good news. Now, there are many factors that might explain these findings, but the timing and quality of the food consumed appear to play a large role. And what's emerging in the data is that people tend to choose less healthy foods during the eating window, so they're not supplying the right substrates to rebuild healthy cells and support basic cell functions. And they're not providing the substrates for energy production or even cell metabolism. Another factor is that certain forms of intermittent fasting don't support our circadian needs. All of our cells perform certain functions at different times over a 24-hour period, and they need nutrients and they need the right signals and inputs in order to do it. And so what the new research is finding is that a 12-hour eating window followed by a 12-hour overnight fast not only supports our circadian rhythm, but produces the same benefits as a longer fasting window. So this is a safer option. And the most updated recommendations come from the work of Professor Walter Longo, who has conducted numerous studies over decades on dietary interventions for those undergoing various treatments for cancer. And really, it appears he's focusing on finding the safest and most effective dietary interventions for those who are the most physically vulnerable to homeostatic changes. But these dietary interventions actually translate to health benefits for all individuals. And his recommendations are to get the everyday diet right and to incorporate intermittent fasting periodically for in the short term, if appropriate. With regards to an everyday diet, the single most important recommendation we can make is to remove processed food. So that basically includes anything that comes in any form of packaging other than, unless it's sea salt, nuts, and healthy oils like olive oil. And because by removing processed food, we're basically left with real food, so food that's nutrient dense and that supports our body's needs. And these foods improve microbial diversity in the species and metabolites that support immune and metabolic function. And as a result, they've been linked to many health benefits, including reducing the risk of cancer and improving recovery for those affected by it. Now, there are certain aspects of the diet that need to be personalized. As I said earlier, everyone differs in their immune response to certain food compounds and also in their metabolic or hormonal response to carbohydrates, and different dietary techniques like different forms of intermittent fasting. So these aspects are best personalized to individual needs. When it comes to periodic intermittent fasting, there is a lot of research into the benefits of the fasting mimicking diet in those undergoing various cancer treatments. This is also the lifelong work of Professor Walter Longo, and it's a diet that is low in calories and proteins. It's a plant-based diet with very specific macro and micronutrient composition, and it's followed 5 days a month. And there are currently 10 human trials underway because preclinical trials found it to be safe and very beneficial alongside cancer treatments. And one of its most significant benefits is improving anti-tumor T cell responses while protecting normal cells and reducing the side effects of the treatment. I won't go into detail, but there's a lot of published data on PubMed if anyone wants to take a closer look at it. Now, when it comes to supplements, the ones that are most commonly recommended for gut health are prebiotics and probiotics, and more recently, postbiotics are becoming popular. Prebiotics are basically dietary fibers that increase microbial diversity and beneficial metabolites. Probiotics are live bacteria that produce a beneficial effect when taken orally, and postbiotics are basically microbial metabolites produced during the fermentation of various foods. Now, of these three, probiotics are most commonly recommended by medical practitioners. But what the research shows is that it's only very specific strains of specific species that produce certain health benefits. But there are a couple of things that we really need to be aware of. Firstly, the benefits are transient and only when the probiotic is taken. Secondly, most of the commercially available probiotics won't necessarily contain these specific strains. And there are only about 40 species that are currently available in probiotic form, whereas microbial diversity refers to having between 160 to 200 species in the gut microbiome. So if probiotics are taken after a course of antibiotics, which generally wipes out our endogenous species, then probiotics will compete for nutrients and attachment sites and can delay or even prevent recolonization and diversity of our natural microbiome. And they also appear to worsen outcomes for those undergoing immunotherapy, whereas a high fiber diet has been found to improve the immune response. So the best recommendations we can make is to increase dietary fiber, because this increases microbial diversity and produces more diverse and sustained benefits. Inulin and fructooligosaccharides have been found to improve immunotherapy response. So these are some of the safe recommendations that we can make. And finally, polyphenols, omega-3 fatty acids, and certain vitamins act as antioxidants and also help to downregulate the inflammatory responses that I spoke about earlier. Some of these, like for example, quercetin, is now undergoing human clinical trials for its ability to remove senescent cells in certain disease states. But with antioxidants, care really needs to be taken. They're not recommended with chemo and radiotherapy because chemo and radio act by producing a pro-oxidant state within cancer cells, so you can't use them alongside, but they don't appear to be contraindicated with immunotherapy. And in fact, omega-3 fatty acids have been found to significantly improve response to immunotherapy in mice studies. So going back to my husband's case, he was started on immunotherapy in February 2019, and he made some really big changes to his diet and supplement schedule. And by July that year, he was left with 2 resolving metastases, which then went on to be cleared by February 2020. And since then, thankfully, his scans have been clear and he was able to return to work 2 years ago. He follows a very healthy diet most of the time, and he does take different supplements on a rotating basis. And we don't know if he just got lucky and we don't know how things will go from here. But given that the microbiome plays such a crucial role in regulating immune function, he will always be focusing on keeping it diverse and healthy. So the key takeaways are basically what we can recommend to our patients to help improve outcomes for any condition. The first one is to remove all processed foods because they're not really food. They're products that change the microbiome and epigenome. And to be honest, we should never have been misled to believe that they're fit for human consumption. Secondly, we can recommend that our patients increase dietary fiber in the form of vegetables and fresh herbs, because the gut microbiome relies on it and our health relies on a well-functioning microbiome. And then finally, for those suffering from any chronic condition, it might be a good idea to consider supplementation as a way of supporting cell functions and restoring homeostasis while the diet is being optimized. Thank you.